Hello everyone, welcome to our presentation titled Exploring Novel AcrIIA5 Variants as Anti-CRISPR Therapy. Design an artificial intelligence-based AlphaFold prediction for targeting a spectrum of Cas9 proteins. This research presents by the Edinburgh Biomakers team. Today, we are excited to share our research journey with you. We'll differ into our exploration of novel ACL2A5 variants as a potential anti-CRISPR therapy, discussing our design strategies and how we are leveraging artificial intelligence through AlphaFold prediction to target a spectrum of Cas9 proteins. Before we dive into our research, let's take a moment to revisit the groundbreaking CRISPR-Cas9 system and the impact of genetic engineering and medicines. CRISPR-Cas9 originated from bacteria as an immunity system and utilized Cas9 protein for genome editing, targeting genetic material from pages. CRISPR-Cas9 then revolutionized gene editing fields, uh, then transcriptional regulation and genome imaging, epigenetic modification, and influence across different fields of biology, for example, genomics, plant science, and animal science. Well, the impact also profound significance. For example, in 2023, the first CRISPR-Cas9 based gene therapy has approved by FDA and UK governments for sickle cell disease and beta thalassemia treatments. It results demonstrating significant efficacy of CRISPR-Cas9 as a drug or therapy. However, with great advancement comes new challenges. We'll address the urgent need to mitigate unintended genome modifications and introduce F5 as a promising solutions. F5 acts as a molecular brake, inhibiting Cas9 nuclease activity and enhancing the precision of genome editing. Look into the pictures. The green picture is F5 3D structure from protein data banks. We obtain this structure and then utilize it as a experiment structure for our research. Then you can look at this picture as well. F5 has ability to bind and inhibits different types of Cas9 proteins like type 2A and type 2C. Our research approach involved harnessing AI through AlphaFold prediction to refine A5 predictions variants. This innovative strategy aims to deepen our understanding of A5 binding mechanism and broaden its implication across diverse Cas9 proteins. The importance of this research is refine gene editing for highly controllable and safety mechanism. Our hypothesis is by using AI prediction can optimize if anti-CRISPR inhibitory potential, broadening its reach across diverse type 2 CRISPR classes. And then The impact is addressing the critical needs for precise control in genome editing technologies, especially in therapeutic applications. We come to the methods. We began by retrieving protein structures that are crucial for our analysis. This involved acquiring the anti-CRISPR-A5 structure and complex structure involving Cas9 from Streptococcus pyogenes, Streptococcus thermophilus, and Neisseria meningitis Cas9s from protein data bank websites. We then extracted the corresponding A5 FASTA data for further protein analysis. Next, we subjected the A5 sequence to BLASTP analysis to identify homologs. We identify a total 67 sequences as A5 homologs and align them using the NCBA multiple sequence alignments tools. Additionally, we perform pairwise alignment on three Cas9 structures to identify sequence variations. Then we select the top 10 A5 sequences and construct a phylogenetic tree representing diverse branches based on these sequences using Mega software. This analysis provides valuable insights into the evolutionary relationship among A5 variants. Then we use AlphaFold protein structure predictions. Our next step involving predictions protein structure using AlphaFolds for the top 10 A5 sequence. We select the top rank prediction and download the structure in PDB formats. We also analyze notable outputs such as percentage of identity, A value, and predicted local distance difference test or PLDDT scores to assess a reliability of the predictions. Next, to predict binding complex between A5 and chosen protein targets, we employ the HDocs server. This involves determining docking and confidence score, which provide insight into anticipated binding affinity and reliability of the docking outcomes, respectively. Lastly, in our investigation, we visualize the alignment of A5 structures and their interaction with protein targets. This was achieved through comparative analysis using visualized tools like PyMols, enabling a comprehensive scrutiny of ligand interaction with Cas9 protein across different species of bacteria. Now let's dive into our research and discussion of our research finding, where we uncover insights into alignment of A5 proteins, comparing Cas9 pairwise alignment, exploring phylogenetic tree of A5, analyzing AlphaFold data, and discuss the implication of our finding. If we look on table 1's top 10 anti-CRISPR homologous A5, we can find that hypothetical protein has a highest query cover for identity and and lowest F values. And then the query cover, all of them almost 100%, except anti-CRISPR protein A5 from E. coli. And then when we talk about percent identity, its span range from 80% into 100% is very diverse. And also, when you look at comparisons of Cas9 pairwise alignments, There are a lot of diverse sequence alignments. We talk about Streptococcus pyogenes and Streptococcus thermophilus. Uh, we know they have a 25% similarities than Streptococcus pyogenes compared to Neisseria meningis. They have around 27 similarities, similar as Streptococcus thermophilus and Neisseria meningis comparison, as well as 27 similarities. Next, we generated a phylogenetic tree using BLASTP, reveals the intricate diversity of A5 protein across top 10 data in different species. The phylogenetic association among A5 orthologs become more evident in figure 2. Notable, the hypothetical proteins from Streptococcus page and A5 protein derived from E. coli display the closest phylogenetic proximity, suggesting a potential shared evolutionary lineage. Additionally, the examination of anti-CRISPR protein in figure 2 uncover noteworthy patterns. The phylogenetic clustering of anti-CRISPR protein A5 from Streptococcus page TP77HL with that from Streptococcus thermophilus implies a common evolutionary origin of conserved genetic elements. Similarly, the grouping of anti-CRISPR protein A5 from Streptococcus sp.wb and anti-CRISPR protein A5 Streptococcus metis exhibiting identical phylogenetics relationships suggest shared evolutionary and functionally convergence within the specific subset of A5 proteins. And then we analyzing the AlphaFold data. We copy paste the sequence into AlphaFold and generate the protein sequences. Then we get the data that reveal several intriguing findings. Notable F55, F5H, and F510 exhibit perfect match with 100% identity with the sequence of the BLASTP analysis, indicating a high level of structural conservation between the sequence from NCBI and the AlphaFold data. On the other hand, F56, while not a perfect match, demonstrated a substantial 89% identity. It's noteworthy similarity in its predictor structure. Further analyzing of range of L value in different orthologs using AlphaFolds explains that A5 to 3, 4, 6, 7, 9, and 10 all exhibit low A values. This range implies robust and statistically significant similarities among A5 proteins. Then A5 become the most lowest A value in this case. And this is emphasizing a exceptionally strong match between query and the database hits. We conduct primal elements that show similar between AlphaFold model A5 variants and the protein data bank structure. Indicate accurate prediction by AlphaFolds. But if you look figure 5, you can highlight diverse conformation and terminal intrinsically disorder region or IDR in A5. IDR flexibility enables broad inhibitory range against various Cas9 homologs. The primary inhibitory function relies on N-terminal IDR especially first 20 residues. If you remove or transient things, reduce A5 activity. AlphaFold's predictor model show low PLDDT for initial 1 until 18 residues of IDR. This indicating local accuracy. A5 variants exhibit mutation in IDR. For example, A5-6, A5H, A510 in the table 2 exhibit a mutation in amino acid in the position 3, phenylalanine, and in the position 10, alanines, and at the positions 20, aspartic acids. This mutation potentially influencing inhibition of distinct Cas9 variants. Let's look into figure 6, the specific residue that impact a functionality of A5 in vivo. We assess the likelihood of A5 model that obtaining a mutation that impact Cas9 inhibition based on previous finding. Based on our alignment analysis, it has been observed there is no mutation that's impact on functionality F5 variants on uh, between structural experimental data from protein data bank and AlphaFold models. These particular residues are conserved and located within a folded regions. It suggests that predicted model of A5 will exhibit functional inhibition of Cas9. Furthermore, if you look on table 3, we examine PLDD score assigned to specific amino acids within the predicted A5 proteins. It reflects different level of confidence of accuracy of structural predictions. Generally, this protein exhibit a high degree of confidence, range from 80 to 90. However, if you look on A5 tens, it stands out due to its remarkable highly PLDDT score of histidine 66, distinct from other A5 proteins. This rating of score reflects varying degree of reliability in structure predictions for the individual amino acids within the A5 proteins. Next, in our study, we employ the S.Web tool for comprehensive docking analysis, exploring the confidence score and docking score of 10 A5 ligands for each Cas9 variants. This approach yields a wealth of dataset resulting of total 3,000 proteins models. From this vast dataset, we selected the two most promising ligands for each Cas9 protein based on this docking score and confidence scores, culminating in 6 exceptional candidates. Furthermore, we utilize PyMol for visualizing these binding interactions, providing valuable insights into the molecular mechanism A5 Cas9 interactions. For Streptococcus pyogenes Cas9, we choose A5 tens and original structural data to binds that have a degree docking score and confidence score, and then we visualize it. Similarly, we use the original structure because it's higher docking score and confidence score, as well as A5 3 that has a docking score and confidence score as well. We omit A5-6 because there is an error in the visualizations because these ligands binds another class of anti-CRISPR rather than the Cas9 proteins. And then we choose in Neisseria meningitis Cas9s, the ligands. The top ligand is the original ones, and then A5 to that have Dawkins score and confidence score that's higher. So that's the chosen ligand that we will visualize in this data. So we came to Streptococcus pyogenes visualizations. In Streptococcus pyogenes Cas9s, a cornerstone of CRISPR-Cas9 proteins, we know there are two nucleases that can have activity to break the DNA, especially NAH and RUVC regions. NAH in the yellow color and RUVC in the pink color. And this picture on figure 7, we know that AcrIIA4, the original ligands, pins on PAM's interacting domains. These regions uh, entirely occupied by A5 molecule, and this region have ability to recognize DNA through interactions between Cas9 prime binding residue. So A4 inhibits the activity of PAM's interaction domains. In other hands, N-terminal IDR and the SCR A5 uh, inhibits REC region. The REC region is important regions because they it has a crucial role in identifying and attaching itself to the CRISPR RNA. So, based on these visualizations, probably the A5 can block REC to interact with the CRISPR RNA and then eventually disrupts the Cas9's system. Then look on zoom figure. In other hands, A5 tends inhibits PAM interacting domain rather than REC domains uh, like original structure. Then it prevents DNA cleavage while allowing DNA binding. This duality of function underscores the diverse inhibitory mechanism probability among A5 in Streptococcus pyogenes Cas9s. Let's move into Neisseria meningitis Cas9s. In this Cas9, the original ligand ACR2C5 interact with the Cas9 at the interface between the wedge and palm interacting domains. It imitates the structure and electric charge of double helix DNA, effectively occupying the DNA binding pocket and hiding the PAM recognition sites. Therefore, the C5, we call it, render Cas9 inactive by obstructing its ability to attach to the DNA. And if you look on A5, yeah, that consists of folded region and terminal IDR, unexpectedly, the inhibitory effect in the A5 of the original structure uh, are observed but an nucleus domain like RUVC and HNH domain of Cas9. Meanwhile, the existing literature depicts various A5 just known to impede functioning of RUVC nuclease domain independently from NAS nuclease domains. Furthermore, A5 AlphaFold models pin RUVC independently without NAH on different orientation to the original structure of A5. The Cas9 RUVC domain specifically cleaves the strand of target DNA that is not complementary. But A5 inhibits the RUVC domain by preventing DNA recognitions, binding, and cleavage. Additionally, it suppresses the function of RUVC domain of Cas9s, regardless of the NH domain activity. Lastly, we talk about Streptococcus thermophilus Cas9s. This Cas9 in the structure has original ligand, which is AcrIIA5 that has capability to obstruct Cas9's interaction through allosteric inhibitions and also has ability to promote formation dimers, notably in PAM interacting domains. Its mechanism of action involves modifying the dynamic Cas9 in relation to PAM bindings. Moreover, A6 we call demise the affinity of Cas9 for DNA bindings, resulting in inhibition of DNA binding within cells. In this is in this research, uniquely, uh, we visualize SCR to A5 based on the visualization of docking process. It can be observed that A5 from original structures engage in interaction and from binding both RUVC nuclease domain and DNA in a clockwise directions. So it can inhibit both nuclease and the DNA. It's so profound and so robust if we can inhibit two uh, structure in the Cas9 proteins. In contrast, A53 only exhibit binding with RUVC, but in a counterclockwise directions. It is highly advantageous for A5 to attain a clockwise position as enable to effectively bind the essential structure of Cas9s. In broader perspective, A5 exhibit the potential to inhibit the activity of RUVC in both Neisseria and Streptococcus casnans. Whereas a case in Streptococcus pyogenes casnan is present two possibilities, specifically the obstruction of REC and PAM interacting. This F5 activity in the Cas9 highlight the diverse mechanism of interaction and shedding light on complexity of CRISPR-Cas9 system. So for the future perspective, F5 potentially being phage therapy against antimicrobial resistance bacteria, serving as an antibiotic alternative. It needs for evidence on its feasibility and effectiveness for this therapy. Secondly, the administration methods for this anti-CRISPR, there are a lot of potential, but wrap it into lipid nanoparticle LNP or adeno-associated virus being one of approach to deliver mechanism of this system. A5 also potentially suppress gene drive when paired with Cas9 ribonucleate proteins. Then further investigation is needed, of course. The third is using directed evolution method to optimize F5 variants for the effectiveness and expand target range. And for the research, we need to validate AlphaFold model through experimental research in the wet lab settings for developing true anti-CRISPR therapy that's effectiveness in vitro and in vivo research. So our conclusion in this research, first, the N-terminal IDR of AlphaFold's model shows mutation, but impedes diverse receptor protein, offering flexibility. A55 identify with a low E value, indicating compatibility as experimental models. The six selected ligands exhibit strong docking score and bind effectively to Cas9 receptor proteins. In this set, ligand hinder RUVC domain in Streptococcus thermophilus Cas9 and Neisseria meningitis Cas9 interact with REC and PAM interacting domain in Streptococcus pyogenes Cas9. A5 adaptability depends on associated receptor protein. It means it depends on the Cas9. And then AlphaFold optimization enables screening of A5 variant structure for specific anti-CRISPR therapeutics. So we need to apply this bioinformatic approach into the real research in the wet lab setting to get more broadened perspective of the A5 activities. Thank you for having me. If you have question, let me know.